Good day, this is the Professor, CBMD PhD, and today's Sunday, February 26, 2023. It is 2:04:19 a.m. Eastern Standard Time, and today's research, microcalcification. So let's hop to it here. Microcalcification, fingerprints, can yield info about cancer. Date February 22nd, 2023. Source, Cornell University. An interdisciplinary collaboration, 10 years in the making, used a material science approach to fingerprint the calcium mineral deposits, known as microcalcifications, that reveal pathological clues to the progression of breast cancer and potentially other diseases. Healthy mineral deposition is a delicately orchestrated process as seen in bone and tooth formation. Sometimes, however, mineral deposits form in places they don't belong, such as kidneys, important example, kidney stones, and breast tissue. In the context of breast cancer, microcalcifications are a critical screening tool because they appear as vivid white specks in mammograms and, in certain cases, indicate the presence of breast cancer. Usually after the initial mammogram, microcalcifications are largely ignored, and what they are saying is they can look beyond the resolution of the mammogram at the microscopic and chemical level and get more information from these microcalcifications. As by taking well-established, high-resolution characterization techniques from materials science and coupling those with an appreciation for biomineralization and how organisms can control the deposition of mineral, we've gained a unique insight into a pathological mineral that they may have important implications for disease. Research groups specialize in biomineralization. Important example, how biological organisms control the growth of crystals in their tissues. More than a decade ago, they began collaborating. And this led to an exploration of a bizarre phenomenon in which bone-like mineral appeared at primary tumor sites. And from there, the collaborators became interested in the way these microcalcifications can capture elements of the tissue microenvironment where they form, almost like a snapshot. The microenvironment, also known as the organic matrix, can in turn influence the mineral's composition, morphologies, and mechanical properties. Minerals have different rules in biology. Minerals forming in breast cancer could be trapping chemical information that reflect their formation environment and that could potentially have clinical value and relevance. As biomineralization is a rather niche area that involves contributions from material science, geology, biology, and more, it is very multidisciplinary, as there is absolutely no reason that oncologists would pay attention to the materials properties of these tiny little crystals that are appearing. We think it really took someone who had an idea of what the mineral could be, offering to do this. They said, can they take everything that they know from studying physiological biominerals and apply it now to these pathological minerals? Next, rather than grind up and homogenize the tissue samples, as other studies had done, the researchers sought to obtain high-resolution, three-dimensional maps of the chemistry of the mineral and organic matrix, such that they would alter the tissue structure. The researchers used a vibrational spectroscopy technique called Raman microscopy that can detect a distinct vibrational signature of a biological molecule's organic and inorganic chemistries, and also map where those signatures are occurring. Researchers then set about integrating and parsing all the data using techniques that were inspired by omics, research in biochemistry, and genetics. One way to look at data when you have a lot of it is to use strategies from the omics communities. It doesn't have to be quantitative, just to visualize how the data behaves. Using hierarchical clustering, they can look at their data as a heat map that gave them an idea of how different parameters that they measured were related to one another and how the different calcifications grew based on their fingerprints. Among the researchers Key findings. Cancer-associated microcalcifications clustered of physiologically relevant groups that reflect tissue type and local malignancy. Mineral carbonate exhibits substantial variety inside the tumor. Trace metals, including zinc, iron, and aluminium, are enhanced in malignant localized calcifications, and the ratio of lipids to proteins within microcalcifications is lower in patients with poor prognosis. While the researchers are not sure if the microcalcifications form before the cancer develops or because of it, the findings indicate there is a correlation with disease severity. The researchers are hopeful the findings may also illuminate calcifications in other types of cancer, such as thyroid and ovarian cancer. The team now plans to study a larger spread of disease characteristics and also apply their approach to other pathological mineralization diseases, such as calcific aortic valve disease, in which mineral forms in the heart valve. The mineral is a disease. And this is the professor of microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.